Hello and welcome to a fresh new episode of Science Monitor, our weekly update on all that is happening in the field of science and technology in and around the country. I'm Aishwarya Kapoor. The names of Nobel awardees of 2015 to curing injections for slipped discs. We are back once again with exciting scientific stories that transpired during the week. Let us take a look at the headlines first. Nobel Prize 2015 announced for Physiology, Physics and Chemistry. Silk-based therapies to treat slipped discs. New clues to stopping bacterial resistance. Researchers trace the origin of extensively drug-resistant TB. In our In Focus segment today, we will discuss about stem cells and their importance in research. And now the news in details. After much waiting and speculations, the names are out. The Royal Swedish Academy of Sciences has announced the names of the researchers who will be awarded the most prestigious Nobel Prize for 2015 in Physiology, Physics and Chemistry. While the Nobel for both Physiology and Chemistry are to be shared by 3 researchers in the field, the Nobel Prize for Physics will be shared among 2. So who are this year's laureates and what are their contributions? Let us see this report.
Nobel, the most coveted recognition in science. The long and eager wait for the announcements of Nobel Prizes for the year 2015 came to an end on 5th October, Monday, with the first announcement of the Nobel Prize in Physiology or Medicine by Erwin Lendal, Secretary of Nobel Committee for Physiology or Medicine. The Nobel Prize 2015 in Physiology or Medicine was jointly awarded to William C. Campbell and Satoshi Omura and Yu too. While Dr. Campbell, a research fellow emeritus at Drew University in Madison, New Jersey, and Dr. Omura, professor emeritus at Kitasato University in Japan, won the Nobel for their discoveries concerning a novel therapy against infections caused by roundworm parasites. Ms. Yu Tu, Chief Professor at the China Academy of Traditional Chinese Medicine, won it for discovering artemisinin, a novel drug therapy for malaria. Following this, the Nobel Prize in Physics was announced on 6th October. The Nobel Prize 2015 in Physics jointly went to Takaki Kajita from the University of Tokyo and Arthur B. MacDonald from Queen's University, Canada. The prize was awarded for the discovery of neutrino oscillations, which shows that neutrinos have mass. While Dr. Kajita was recognized for the discovery that neutrinos from the atmosphere switch between 2 identities when coming to Japan's Super Kamiokande neutrino detector. Dr. MacDonald and his team has demonstrated that the neutrinos from the Sun, instead of disappearing on their way to Earth, were captured with different identity when arriving to the Sudbury Neutrino Observatory. The Royal Swedish Academy of Sciences also announced the Nobel Prize in Chemistry for 2015 on 7th October. The Nobel for Chemistry is jointly awarded to Dr. Thomas Lindahl from Francis Crick Institute and Clare Hall Laboratory, UK, Dr. Paul Modric, Howard Hughes Medical Institute and Duke University School of Medicine, USA, and Dr. Aziz Sankar from the University of North Carolina, USA, for deciphering the mechanism of DNA repair.
Painkillers, exercises and operations. As many of us would relate to, all these are just temporary reliefs when it comes to back pain caused by slipped discs. Now for patients looking for a permanent cure for slipped discs, there might soon be an answer in the form of engineered discs and injectable hydrogels. Now a team of researchers from IIT Delhi have developed 2 special approaches based on silk, which may be adopted to restore the cartilage in the spines, thus curing slipped discs. Let us see this report.
With changing lifestyles and sedentary work environments contributing to bad posture and stress, It is estimated that 3 out of 4 Indians suffer from back pain at least once in their lifetime. While the painful condition of degenerated intervertebral disc is currently treated using surgical procedures and artificial metallic implants, their benefits are short-lived, accompanied by post-operative trauma. But soon we may have better technologies to cure the problem. The solution comes in the form of injectable hydrogels and silk fibrous scaffolds, using the silk and human cells for regenerating the cartilage. The approaches use silk fibers extracted from the cocoons of Bombyx mori and have been developed by a team of researchers from the Indian Institute of Technology, Delhi, under the guidance of Dr. Saurabh Ghosh.
So we are following two different strategies. In one strategy, we are developing scaffold to replicate the anatomical alignment of collagen fibers present in intervertebral disc, and for that our favorite biomaterial is silk. We are first studying the alignment of collagen fibers present in intervertebral disc of our spinal joint, and then we are wrapping silk fibers in a similar manner to replicate the crisscross pattern of collagen fiber present in intervertebral disc. In our second approach, we are trying to develop injectable silk hydrogel, where we are first developing microparticles and encapsulating glycosamine as a drug molecule, and then we are mixing with patient's own stem cells, adipose tissue derived stem cells. Then we are mixing it together in silk hydrogel to develop injectable silk hydrogel.
While the first approach involves the development of silk scaffolds on which cartilage tissues can be engineered, the second method makes use of silk hydrogels. The team has developed a special silk scaffold support system on which cartilaginous tissue can be grown. These specially engineered scaffolds resemble the anatomical alignment of collagen fibers in the human spine and can be used as an implant to treat the degeneration of the intervertebral disc. According to the researchers, not only to treat IVD, the silk scaffolds developed by the team can be specifically engineered for various purposes, ranging from creating random porous scaffolds to mimic the bone structure to textile breeds.
Silk is a promising material for tissue engineering applications owing to its superior mechanical properties, its toughness and resilience compared to other polymers available. So as the entire amino acid sequence of silk protein is known, therefore it is easy to chemically decorate it with additional compounds such as diazonium coupling or decorating it with cell adhesion motifs, which can further improve cell compatibility on the surface of silk.
The study has been published in the journal ACS Applied Material Interfaces. While this procedure is invasive, the same team has developed a non-invasive approach that makes use of injectable microspheres of silk protein, fibrion. The technique makes use of fibroin microspheres filled with a drug called glucosamine. Glucosamine is currently used to treat osteoarthritis and is involved in cartilage production in the spine. The microspheres embedded in a silk hydrogel along with stem cells of the patients and injected into the degenerated discs restore degenerated cartilage. The study has been published in the journal Biomaterials.
Basically, our system works by the action of glucosamine, a drug that has been earlier used for the treatment of osteoarthritis. We thought of this system to deliver a controlled release of this drug into the degenerated intervertebral disc, so that a slow healing property of this drug can be taken advantage of. So when we put this system into the body of the patient, the drug glucosamine is slowly released from the microspheres that helps in generation of extracellular matrix molecules and helps in the healing of the disc.
While both the techniques have so far shown promising results, researchers are now planning to take up the trials and preclinical studies.
Extensively drug resistance or XDR-TB, a life-threatening form of TB which cannot be cured by existing drugs. Caused by a mutated strain of TB bacterium, XDR-TB is being reported worldwide. Now, in a path-breaking discovery, researchers working on tackling the issues of drug resistance have traced back the origins of XDR-TB to KZN province in South Africa. Well, the discovery holds great promise for finding novel drugs that may stop bacterial resistance.
Tuberculosis, an affliction that still kills millions worldwide every year. First multiple drug resistance or MDR and then extensively drug resistant TB or XDR-TB. With the emergence of drug resistant strains of tuberculosis bacteria, the threat has increased even more. While the medical world is cringing in fear of XDR-TB, which cannot be treated with any of the currently available drugs, researchers have traced the origins of the deadly strain. Based on a study conducted by scientists from the KwaZulu-Natal Research Institute for Tuberculosis and HIV, the Broad Institute of MIT and Harvard in the US, the roots of XDR-TB can be traced to KZN or KwaZulu-Natal Province in South Africa during the late 1950s. Researchers have sequenced the genome of XDR-TB strain and has determined that the special mutation that gives it drug-resistant properties to TB bacteria first emerged in 1957, soon after the first antibiotics to treat TB became available.
They have done the sequencing of genome in resistant strains which they have found during that outbreak or presently, or some strains we preserve in the past also in 80s. They have taken those strains also and they have done the sequencing. By sequencing we know the genetic material, what is the sequence of genes in them in genome, when the mutation took place, and we can do dating also, at what time, how many years back these changes have developed. So they say whenever there is an infection, because strains are coming on through the ages, through different years, they have already developed to the resistance to the drugs which we use nowadays. So we have to take precaution whenever we get a patient of TB. We should get resistance pattern to know to what drugs the patient is resistant, and we should tailor-made the schedule for that patient according to the resistance pattern.
According to the study, the ordinary strain of TB bacteria underwent mutation to become multi-drug resistant by the 1980s, and by the mid-1990s, it mutated to become extensively drug resistant or XDR. During the study, researchers also discovered that the first drug all the strains developed resistance to was the antibiotic isoniazid. And this was the crucial step that started the evolution towards XDR. Non-compliance to the prescribed dose of TB drugs by patients is known to be the main reason behind the emergence of MDR and XDR-TB. Although the researchers were tracing the evolution of Tugela Ferry XDR strain, many other strains of drug-resistant TB that were evolving independently of each other were also discovered during the process. While it is believed that problem of XDR-TB, especially in South Africa, is due to recent failings in the TB control program along with the explosive HIV epidemic, the study that has been published in journal PLOS Medicine indicates that resistance had actually started evolving since the introduction of the first antibiotics to treat TB. And once a resistance emerges, it evolves and continues to be transmitted. The study has important implications towards understanding how XDR strains spread, as well as for the management and treatment of TB across the world, along with the development of novel and more effective drugs.
And now it is time to take a short break. We'll be back with more science news. Keep watching Science Monitor. Welcome back after the break. You're watching Science Monitor. Let us now have a look at some important science and technology activities happening in India and abroad in our next segment, Science Express.
India will soon join the League of Nations who owns the space radar technology. The declaration came from former chairman of the Indian Space Research Organisation, Dr. Madhavan Nair, while speaking at an event in Kerala. The space radar will be used to prevent the collision of satellites and spacecraft, as instructed by United Nations Committee. The event was attended by students from various schools who enthusiastically participated in the innovative demonstrations made to encourage students about space technology. In a proud moment for the nation, Indian-American researcher Dr. Karthik Chandran has been named a 2015 MacArthur Fellow with the Genius Grant. Dr. Karthik Chandran is an Associate Professor of Earth and Environmental Engineering at Columbia Engineering. He has been recognized for his work in transforming wastewater from a pollutant requiring disposal to a resource for useful products, such as commodity chemicals, energy sources and fertilizers. Dr. Chandran, under the Genius Grant, has been awarded a stipend of $6,25,000. With the objective of fully immunizing children below 2 years in the country, Union Health Minister J.P. Nadda on Monday launched the second phase of Mission Indradhanush. The second phase, which is to run for a week, aims to vaccinate children who are either under-vaccinated or partially vaccinated against 7 diseases which include diphtheria, pertussis, tetanus and hepatitis B. The week-long program is to be implemented in 352 districts of the country. In another major step towards creating a cleaner and greener environment, India has unveiled its climate target to cut carbon intensity, preparing for the UN summit in Paris. In December, India has pledged to make its economy more energy efficient and cut down the carbon produced per unit of GDP growth by 33 to 35% by 2030. India has also targeted generation of 40% cumulative installed power capacity from non-fossil fuel sources by 2030. The nation is also aiming to create an additional carbon sink of 2.5 to 3 billion tons of CO2 equivalent through additional forest and tree cover by 2030. Mr. Bishnu Tamang, a local from Raghamohan village of West Bengal, has devised a novel warning system to alert villagers of elephant attacks in villages near forest areas. The early warning system comprised 2 switches fitted with a nylon thread with a doorbell attached that run on 2 batteries. As soon as the elephant comes in contact with the nylon thread, alarm goes off. The technique has been installed in fields and has been found to be effective is warding off elephants that destroy crops.
It is indeed amazing how a single fertilized egg cell can assemble itself into a fully functional human body. It would be interesting to note that all the cells in our body, from heart cells to neurons, bone cells to skin and blood cells, which widely differ in their structure and properties, are derived from a common mass of cells called stem cells. Now, therapies based on stem cells are today a frontier research area offering wide scope. So, what are stem cells and why are they important? Well, this would be the topic of our discussion in our next segment, In Focus.
Human body is perhaps the most complex machinery that was ever assembled. From head to toe, human body comprises various organ systems made of tissues, each designed specifically to carry out a specialized function. A skin cell is different from the cells of the heart, which is quite different from the brain, which in turn differs from the tissues of the intestine and kidneys. Ever wondered how a single-celled egg, when fertilized, can give rise to such huge varieties of differentiated cells? Or how our skin or blood tissues get replenished as the old ones die? The answer is stem cells. Stem cells are a special class of undifferentiated cells found in mammals that has the ability to divide and differentiate into specialized cell types. These special cells possess the amazing capacity of self-renewal, as a result of which they can divide any number of times while maintaining their neutral, undifferentiated state. At the same time, they are totipotent or pluripotent, which means they retain the capacity to specialize and differentiate into specialized cell types as required. The history of stem cells is quite recent and can be traced back to early 1990s, when attempts were made to fertilize mammalian eggs outside of the human body. Then it was discovered that some cells had the ability to generate blood cells. In 1978, stem cells were discovered in human cord blood. In 1981, first in vitro stem cell line were developed from mice. Embryonic stem cell lines were created from a hamster in 1988, and in 1995, first embryonic stem cell line was derived from a primate. A major achievement came in 1997 when a lamb was cloned using stem cells. Experts categorize two types of stem cells found in humans. The first class, known as embryonic stem cells, are derived from the inner cell's mass of 4 or 5-day-old human embryo, that is, in the blastocyst phase of development. These are the stem cells that divide and differentiate to develop into a full-formed baby. The second class, known as the adult or somatic stem cells, are found in specific tissues of adult humans, such as brain, bone marrow, blood, skeletal muscles, skin, and the liver. It is generally believed that while embryonic stem cells has the ability to differentiate into any kind of cell, the adult stem cells are limited in their ability to differentiate based on their tissue of origin and remains dormant until activated by disease or tissue injury. With the advent of technologies today, stem cell research has progressed dramatically and countless research studies are published each year on stem cells. Today, stem cells are extracted from adult tissue, umbilical cord, blood cells, or from a dividing zygote. The cells are cultured under controlled conditions in laboratories, where they divide to form healthy, dividing and undifferentiated cells called the stem cell lines. The stem cell lines can then be instructed using special signals to specialize and induced to form specific type of cells. Today, cord blood banks are gaining fast popularity. Umbilical cord blood collected after childbirth contains stem cells, which can be used to treat hematopoietic and genetic disorders that developed at a later stage in life. Researchers across the globe are developing cell-based therapies to treat disease using stem cells. The novel area is referred to as regenerative or reparative medicine. Regenerative medicine involves use of human stem cells to regenerate cells and tissues, which can be then used to treat various degenerative disorders including muscular degeneration, Alzheimer's, spinal cord injury, stroke, burns, heart diseases, diabetes, blindness, Parkinson's, osteoarthritis, and rheumatoid arthritis. And what is more, sometime in near future, scientists may even be able to grow tissues and organs in Petri plates, which can be used for transplant in place of donated organs and tissues. While stem cell research offers huge potential, the studies also come with many limitations and ethical concerns. While extraction of stem cells from cultured embryos is considered unethical by many, stem cell therapies also requires immunosuppression if the cells are used from different individuals. But with intense research on the way, we can soon expect stem cell-based therapies to come to the rescue of humans from many disorders.
Well, that is all for this episode of Science Monitor. Do tell us how do you like our program. You can send your feedback and suggestions. Our email ID is news@vigyanprasar.gov.in. You can also write into us at Vigyan Prasar C24, Qutub Institutional Area, New Delhi 110016. Well, that is all for today, but we'll be back with fresh news stories on science and technology again next week. Till then, keep watching Rajya Sabha TV and think scientific. Bye-bye.